तेरी तो ले गया इसने, कुछ तो बोला है, तू बोला था सुबह।
So, good afternoon, everyone, and I am Rishabh, and I hope I am all of you are able to see my screen. So, let's start for today's lecture. So, today we'll be discussing about the content that you have studied in week 2. Okay, so starting with the first question. So, in culture media, dash is used as a source of which compound for glycolysis? So the options are carbon, glucose, nitrogen, ammonia, glucose, carbon, or lactose, carbon. Can anybody guess?
Option C, sir.
Okay, any more answers?
I also think option C.
Okay, yes, the answer is option C. So glucose is used as a source of carbon for glycolysis. Okay, and in case of nitrogen source, we use certain amino acids like glycine or alanine for the as a nitrogen source, and that is used for the nitrogen requirements of the cell. So moving on to the next question. Which of these can be a low-cost substitute for a separate sterile room? So here are the options. Can anybody guess?
Option C, sir.
Okay. Any more? Uh, so yes, the correct answer is option C. So, like for a cell culture room, we need a separate space so that we can grow the cells under sterile condition. So, what we can do is like if we have to maintain a cell culture facility, we have to grow cells in a separate room that will have certain things that are required for the cells to grow. For example, there will be an incubator. There will be some storage compartments also that can be -80s or the refrigerators that we use. So now if we see, we cannot grow these culture like if we require a big space, and like the most of the people, depending on the funding and depending on upon the what work we do, it requires a lot of area. So a small substitute for that full room is a laminar flow hood, where we can transfer culture, or we can do any number of experiments related to cell in that hood. So the correct option is option C, that is laminar flow hood. Okay, so let's move on to the next question. So this is the basic diagram of a laminar flow hood that we use in our cell culture systems. So there is a inflow of air, and that basically filters the air out. The inflow of air that filters the impure air that is coming from outside, and it passes through a filter that allows only pure air to pass through. And this is the working bench where we can keep our cells and all the materials that we are using. And there, this is a particular cell culture hood, and there are different kinds of hood depending upon the culture that we are doing. So in case of a bacterial hood, we will have a different set of requirements there, and there will require a, a source of heat also. So in that case, we use a Bunsen burner and all those things. And like we also have a lamp here. We also have a lamp here that will be uh, both a normal light as well as a UV light. UV light we require for sterilization. So before starting any experiment, we just switch on the UV for some minutes, basically 5 to 10 minutes, and then we start our experiment by wiping the hood with some uh, sterilization materials. Okay, so if anybody has any doubt, can ask. If not, then we'll move on to the next question. So this is a general curiosity question. I hope everyone would remember some of the basics of the gas equations and all those stuff. So the question states 1 liter of liquid nitrogen generates nearly dash amount of nitrogen gas. So here is the information that I have given. You might require these things in your calculations. So please. Yes. Mhm. I request everyone, please mute yourself. If you have any questions, you can ask by unmuting yourself. So please try this question. We'll discuss in 5 minutes. So can anybody try doing it? Like, how will you start solving this equation? Okay, I'll give one hint for people who don't know, who don't remember. So there is an ideal gas equation called as PV equal to nRT. Okay, so you can use this equation to calculate the amount of gas. You can see. Okay, can you explain how did you do it? So please apply this formula and then we'll discuss. So here what we'll do is, this is the ideal gas equation that we know. So P stands for pressure, V is the volume of the gas, N is the number of moles of the gas, R is the gas constant, and T is the temperature. Okay. So here we have the density of the liquid nitrogen that we have, and from that we have to calculate how much nitrogen gas will be produced from that particular amount of liquid. Okay. And then
So we have to find the
so from this information, we have all those things. We have direct value of gas constant, temperature, and pressure, and we have the density of liquid nitrogen. So from this density, we'll calculate the number of moles that we have.
N
So
is equal
for
to M by M. We will calculate volume. Then finally we get substitute the values. Then 0.809, 2.8, all those values, and we will get finally the volume.
yes, yes, yes. So for N, we have the number of moles. So the definition of mole is the given mass that we have upon the molecular weight. Okay, and molecular weight of liquid nitrogen, that is N2, in all cases is 28. Okay, so from this we need the given weight, and that we'll get from the density into the volume. So density states that mass upon volume. So it's mass per unit volume. So for mass, we'll get the value as density into volume. Okay. And here we have the density of liquid nitrogen as 0.807 gram per ml, and volume we have is 1 liter. Okay. So we'll substitute these values here, and then we have to calculate the volume of air that is evaporated from this amount of liquid nitrogen that we have. Okay, so now this volume is equal to density into volume into R T by P into 28. Okay, so now after substituting these values, we'll get a value that is near to 700 liters. So this will give you the answer, option C. So anybody has any doubt in this case, can ask.
Sir, can you repeat the explanation once again?
Yes. So I hope you know the ideal gas equation. PV is equal to nRT that we use to calculate the variables in case of any ideal gas that we consider. Okay. So P pressure, V is volume that we have to calculate, N is the number of moles, R is the gas constant, and T is the temperature. Okay. So now we have the values, direct values of gas constant, temperature, and pressure. Okay, but we need the number of moles of gas that are present. So for that, we have the density of liquid nitrogen as this given 0.807 gram per ml, and the volume of liquid nitrogen that we have is 1 liter. So from that, we'll calculate the number of moles. So for that, we need number of moles is equal to the given weight of the gas upon, divided by the molecular weight of the gas. So that is given by N2, that is 28. So this is the molecular weight of liquid nitrogen. And for M, we have density as well as the volume, because density states that it is mass per unit volume. Okay, so for to calculate given mass, we'll multiply density into volume. So density is this thing and volume is 1 liter. So this will give you the given mass of the gas. Okay, so now we have to calculate the volume of liquid nitrogen. So for that, we'll rearrange this equation and this will lead you to this formula. So this will be density of liquid nitrogen into its volume into gas constant into temperature, that will be divided by the pressure and the molecular weight of the gas. Okay, so after substituting all these values, it will yield an answer of nearly 700. So it will be 705 or 706. So that will be nearest to 700 liters. So that's why this will be the answer. Okay. Everyone understood the question?
Yes, sir.
Okay. So let's move on to the next question. So which of the following is used for sterilization of the laminar flow hood? And these are the options. Can anybody guess?
Alcohol.
Okay. Any more
Alcohol.
answers? Yes. So the answer is alcohol. And before starting any of the experiment, we just wipe the hood with alcohol. That is 70 to 75% alcohol we use generally in our labs. So we just wipe the whole hood, the working area, and then we switch on the UV.
We're using ethanol.
Sorry, can you?
Is ethanol is used for sterilizing of food?
Yeah, generally ethanol is used. And then after that, we can switch on the UV for some time, or we can switch on the UV previously and then wipe it with alcohol. So that allows the any contaminant or any other thing that is lying on the working bench can be cleared out. And this is the main use of alcohol or our sterilization before working for any experiment of the group. Okay, so let's move on to the next question. So higher level of oxygen in cultures can be toxic due to more solubility of oxygen in medium, depression in free radicals, drop in freezing point, or increase in free radicals. Can anybody guess?
I think it is option D, increase in free radicals.
Okay, any more answers?
Sir, I think it is more solubility of oxygen in the medium.
Okay. Any more answers? So, what happens is in normal scenario itself, when we consider, so this is our cell, and this is the mitochondria that we have. So it's the powerhouse of the cell. So what happens is this is the main organ which utilizes oxygen. Okay, so it also releases certain reactive oxygen species, or we call it as a free radicals that are toxic to the cells. So that's why there is oxidative stress in the cell also. So what happens is in a cell culture where we are growing cells, if there is more amount of oxygen in case of those cell cultures, there is more free radical production or more Ross generation that we see. So this is the main reason that it is toxic to the cells, because it increase in the free radicals. Okay, so anybody have any doubt in this case?
Sir, what do they do, the free radicals?
So free radicals generally what they do is they like the major outcome is they will do damage to the DNA, okay, or any other membranes that they are in. So if the levels of free radicals increase above a particular threshold level, they start damaging the nearby proteins, or they'll interfere with all the electrical or all the charge related processes that happens in the cell. So that is why they are toxic to the cells. And it also increases the oxidative stress that we call in our physiological language. So increase in free radicals increases the oxidative stress inside the cell. So shall we move on to the next question? Okay. So the next question is, which of the laminar hoods does not throw air at the person performing experiments? So the options are horizontal laminar flow hood, vertical laminar flow hood, both or none. So can anybody guess?
Option B.
Options
I think
B.
option C,
Okay.
sir.
C. Okay. Any more answers? The correct answer is option B. So these diagram explains the working of both the laminar flow hood. So what happens is, in case of horizontal laminar flow hood, there is direct supply. Suppose this is the working bench. Okay, so the number 4 is the working bench. So here the person is sitting and he is performing the experiment. So what happens is, this is the back of the flow. Okay, so from here the air will enter and it will pass to the filter, and then it will go on to uh, the other side and release out from the other side, making a sterile environment in this space. So it forms a laminar flow in this place. But in case of a vertical laminar flow hood, what will happen? Here, the person will be sitting and doing the experiment, and from above, the air will pass and it will maintain a laminar flow, and it will release from the back end or the downpour. So here there will be some system to release the air from from below. So it will not directly pull the air on the person who is working with the who is performing the experiment. So it will not directly pull the air on the person. But in case of a horizontal laminar flow hood, the air will be directly pulled on to the person that is sitting in front of the. So the answer would be B. Okay. So any doubts?
So then can we say that vertical laminar fluid is more safer while working with infectious specimens?
Yes. So vertical laminar flow hood will be more safer in case of when we are working with more infectious agents. So for DSL-3 and DSL-4, generally vertical laminar flow hood is preferred.
Thank you, sir.
So any more questions related to this? If not, then we'll move on to the next question. So the next question is sterile liquid should be stored at which temperature? Can anybody guess?
Room temperature.
Okay. Any more answers?
Option B.
Okay.
20 to 25 degrees centigrade. That is room temperature.
So yes, the correct answer is room temperature. And any sterile liquid that is there. So when we make buffers or any other thing that we store after sterilization or autoclaving, we generally store it at room temperature only, if specified. So generally, most of the reagents or the other things that we use that are also sterile itself and that we do not sterile after making or something. So those things we store at different temperatures according to their stability. Okay, so we'll move on to the next question. Which of these is the most central organs of the cardiac system? So I think everybody will know this.
Option D. Heart.
Yes. Yeah. So the correct answer is option D. So yes. So most cell lines grow well at which pH? Can anybody guess?
Option C.
Option C.
Yes, the correct answer is option C because this is the physiological pH that is maintained in our body also. So most of the cell lines that are isolated from animals or animals are grown at this particular pH. That is 7.4. Okay, so yeah. Which of the following components of cell culture media are matched correctly with the function they perform? So option A, phenol lead, it regulates osmotic pressure. By option B, bicarbonate, it's a pH indicator. Option C, inorganic salts that are helps in regulation of osmotic potential and membrane potential. Or option D, it's glutamine that is a pH indicator. So can anybody guess?
Option C. Inorganic salts.
Okay, any more answers? So yes, the answer is option C. So in this case, phenol red that we use is used as a pH indicator. Okay, it's not used in the regulation of osmotic pressure. Bicarbonate ion is used in buffering for its buffering activities. So it's used as a pH buffer. So yes, inorganic salts is used in regulation of osmotic pressure and membrane potential, and glutamine is used as a source of sugar as well as amino acids. Okay, so this is these are the basic things that we require in a compound in a media. So first is the buffering system that helps in regulation of pH. So natural buffering that we use is CO2 that we studied in the last week. And then we use different buffers like trace or HEPS buffer that maintains the pH around 7 to 7.4 in a cell culture. Then we use a pH indicator. So this is the most important component of the media because this only tells us that whether the cell is metabolizing or not, or there is any contamination or not. So if there is any contamination, then the pH of the pH will change, and that will change the color of this pH indicator. So we use this as a pH indicator. Then the inorganic salts that we have they retains the osmotic balance and the membrane potential also. So we use different salts like sodium chloride, potassium chloride, and as well as some calcium salts for different cell lines. So the inorganic salts used for this purpose. Then for amino acids, we require a source of nitrogen as well because the cells are actively growing. So they require some amount of nitrogen for the production of proteins. So for synthesis of proteins as well as proliferation of cells, amino acids is required. So we generally add glutamine and it provides nitrogen for NAD, NADPH and formation of nucleotides because there is nitrogenous bases present in that, as well as some secondary metabolites that the cells require. Okay. And carbohydrate, they are the main energy source that we discussed just now. So glucose and galactose are generally used, and in some cases, maltose and fructose are also used. And then in the last thing, we require fatty acid and lipid. They are particularly important in serum-free media that we'll be discussing in the next week. And there is one more thing that are antibiotics that we use so that any other contaminant or any other cell line, any other cells that are not belonging to that particular cell line does not are not allowed to grow in that particular media. So antibiotics are also used for this in this media. Okay. So anybody has any doubt in this question? So if not, then we'll move on to the next question. So different oxygen concentrations are needed for replicating different in vivo conditions. So we want the oxygen levels in percentage that are maintained in these types of conditions. So can anybody tell what is the range of oxygen that is maintained in the normoxic conditions?
Twenty to 21%.
Sorry, can you be a bit louder?
20 to 21%.
Okay. Any more guesses? So yes, for normoxic conditions, we generally use from 18 to 21%. That is the range of oxygen that is maintained in normoxic conditions. Okay. So, and for hypoxic, can anybody tell?
Less than 90%.
Okay. Any more answers? So generally hypoxic conditions is for hypoxic condition, the level of oxygen that we maintain is nearly 5%. Okay, less than 5% rather. So nearly 5% is the oxygen concentration that is maintained in the hypoxic conditions. And for anoxic conditions, can anybody answer?
Absence of oxygen, 0%.
Okay. Any more answers? So in case of anoxic conditions, we require a concentration of less than 1%, less than equal to 1%. That is the oxygen concentration maintained in anoxic conditions. Okay, so if we have to maintain any of the conditions that are uh, in vivo present. So normoxic conditions are present normally in the human being. Okay, so every organ now you are sitting. So that are normoxic conditions that are present in the human being. And hypoxic is the condition that are related to generally tumors, because in case of solid tumors, there is a microenvironment maintained at hypoxia. So that allows for tumor progression and metastasis as well as angiogenesis. So in some cases, we need that hypoxic condition to be replicated in cell culture systems. So we maintain hypoxic conditions there, as well as for some other processes. And anoxic conditions is also one of the conditions where there is no oxygen concentration, especially when we are studying the role of uh, certain molecules that are regulated by the oxygen. So there we need anoxic conditions so that that is maintained in less than 1%. So these are the different ranges of oxygen that we need and that are maintained in the cell culture system. Okay. So anybody has any doubt?
Sir, can you repeat this this one explanation again?
Okay. So what do we do is like the normoxic conditions are maintained inside our body, like in general case. So if we are currently we are breathing normally and we are in a normal oxygen levels inside like outside our cells. So that is the normoxic condition. So we have to replicate that in for in cell culture system also. So we need around 18 to 21% of oxygen percentage that is maintained in the cell culture system. So this is the normoxic case that is generally maintained in the cell culture system. And this is the hypoxic case. What happens is, like I'm taking an example of solid tumors. So what happens is in case of a solid tumor, there is hypoxic environment maintained in the environment of the tumor. So that helps in the progression of that tumor as well as the metastasis and angiogenesis of that particular tumor. So to study that in vitro, we maintain a hypoxic condition that is less than 5% of oxygen is present in that case. Okay. And the third one is the anoxic conditions, like it's nearly no oxygen. So that's why it's the name anoxic. So we have to study some certain things in the development or the role of oxygen in development, then we can use anoxic conditions in that case. And that's less than 1% that we use in case of our cell culture system. Okay. So am I clear or any doubts? If not, then we move on to the next question. So yes, so this is a calculation based question and we have to apply simple maths in this. So, and this is related to cell counting as well as the cell seeding that we discussed in the last lecture. So the question states the total number of cells in a culture is counted using the Trypan blue exclusion assay. So this is a particular assay used to tell whether the cells are living or not, and is found to be this value 2.7 into 10 to the power 6 per ml, cells per ml. And then the culture was diluted 1 in 27 times, and then 100 microliter of the dilution was used to seed every well in a 96 well plate. So what is the cell density or the number of cells present in each well after adding the 100 microliter of the diluted solution? So can any please try this question and then we'll discuss. So has anybody got any answer?
So option A.
Option A. Okay. Any more answers? Okay. So how will we solve this question? This is simple maths, nothing else. Just we have to uh, know what we are applying. Okay. So we know that the suspension that we have of cells is have 2.7 into 10 to the power 6 cells per ml. Okay. And from this suspension, we diluted it at 27 times. Okay, so now the final concentration after dilution at 27 times, the concentration becomes 1 into 10 to the power 5 cells per ml. Okay, so this is the first point. So from the crude suspension that we had, that we isolated from the particular plate, is this much, and after diluting that 27 times, we got the number of cells that are present in the diluted suspension is this. Okay. And after that, what we did was we took 100 microliter of this suspension and we poured it in each well of the 96 well plate. So for this well that we added 100 microliter, so 100 microliter into 10 to the power 5 cells that we had in 1000 microliters. Okay, so 1 ml is equal to 1000 moles or 1000 microliters. So when we calculate this from this particular equation, it comes out to be 1 into 10 to the power 4 cells that we have in each well of the 96 well plate. Okay, so the answer is 10 to the power 4 cells that we have in each cell or in each well of the 96 well plate. So am I clear? Are there any doubts in this question? So this question can be asked in other way also. Like if you are seeding in a 6 well plate and 1 ml of the media, 1 ml of cells are added to each well, and this is the cell density, then what's the dilution that you are using? So this question can be asked in other way also. And secondly, this question is important in your every cell culture experiment because like when we are seeding cells for a particular experiment, for example, if you are seeding in a 24 well plate and you want around 30,000 cells to be present in each well of the 24 well plate. So you have to calculate the number of cells that you had from a plate that you are seeding as well as the dilution that you make for cell counting as well. So we'll use a hemocytometer for cell counting that we discussed in the previous lecture. And from that, we'll calculate the number of cells that we have. And then from that, we'll add the particular volume of cells by calculating that, okay, when if we have to add 30,000 cells in one well, then we'll add this much amount of cells from the suspension in that well. So that will yield 30,000 cells in that particular well. So this particular calculation is very important in your day-to-day cell culture experiment. So are there any doubts in this question? If not, then we'll move on to the next question. So yes. So what will be the color of phenol red in cell culture media if there is a bacterial contamination? Can anybody answer this?
Option C.
Option C. Okay. Any more answers?
So option C.
Okay. Any more answers? So what happens is when phenol red is at neutral pH, so that is around 7. So 7.4 is the physiological pH that is maintained inside the cell culture. So what is the particular color that it gives in the media? Can anybody tell?
Dark pink.
Dark pink, or rather we say that it gives a red color to the media when it's at a physiological pH. Okay. And when it's metabolically active. So as we know, when the cell is metabolically active, so it secretes certain compounds that makes the media acidic. Okay. So here the when the media becomes acidic, that is around 5 to 6, the pH of the media will become 5 to 6. So here the culture will become yellow or orange. So when there is some acidity, sorry.
So if
Mhm.
if it is more acidic, then it will turn to yellow or orange.
Yes.
Okay, sir.
So when the cell is metabolically active, or when it grows and proliferates, or the number of cells increases. So in that case, the cells require more of the metabolic activity to be produced, more glycolysis, more energy production. So that in that case, the media will become acidic due to release of the toxic compounds, and in that case, it will become yellow or orange. Okay. Now, if there is any contamination present in the media, specifically fungal or bacterial contamination, so what happens in that case? The media becomes alkaline. Okay, so this pH of the media becomes from 9 to 10. So that case, the media turns pink. Okay, so in case of contamination, the media will turn pink, and because of the rise in pH due to contamination. And it will also give a peculiar smell that is not usually comes from the normal cell culture. Okay, so this is the scenario that happens inside the cell culture in case of a pH thing. So the correct answer is option D. So in case of a bacterial contamination, the culture media will become pink instead of red. So this is the explanation. So yes, that is what I was explaining to you guys. So in case of a metabolically active cell, the media will turn around yellowish orange kind of thing. And this is the normal pH. This is dark pink, or rather we say red. Okay, depending upon the components of the media, some things also happen. So this is dark pink or red, and this is pinkish color that happens due to contamination. And it also increases the turbidity of the media also. Okay. So any doubts in this question?
Sir, how does the contamination make the media alkaline, sir?
Okay. So I think like in the next weeks, we'll discuss more about the culture media, then it's better to explain those things better. Okay, because there are certain technical stuff that are involved and other components of the media are also involved in that process. So I'll explain you these things again in the next week also. So there you will get an answer. So any more doubts? Not, then we'll move on to the next question. So this is related to the biosafety levels that we study for different organisms. So a person is cloning plasmid DNA in E. coli DH5 alpha strain. So which type of biosafety level cabinet does he working in? So BSL 1, 2, 3, or 4. Can anybody guess?
So option B, BSL2.
Okay. Any more guesses or answers? So the correct answer is option A, that is BSL1. So here is the explanation. So what we do is we generally from as we rise from BSL1 to BSL4, this is the increase in risk with the microbes or with the organisms that we are handling. Okay. So for BSL1 that we use is the organisms that are not harmful to the humans. Okay, and it will not cause any potential laboratory hazard or any outbreak. So that's why we call it as BSL1 and all the non-infectious bacteria that we are working with. So in this case, we are just cloning any plasmid DNA in E. coli. So E. coli that we know that particular strain DH5 alpha is non-infectious. Okay, so here we'll be working with BSL1 only. And it's in day-to-day experiments also that we work on, or the laboratory, normal laboratory that we have used till now is BSL1 for cloning, or the normal bacterial culture that we have studied. Okay. And in case of BSL2, the hazardous potential increases a bit. That's why it belongs to the next category or the higher category. So the organisms that are moderately hazardous or to the personnel or the environment. So that can be included in BSL2, and most of these things can be worked in BSL2. So the non-infectious things that we are working with can include in BSL2, for example, hepatitis A virus, because it is not uh, like uh, it is not uh, communicable. So it is it is transmitted through food or any other thing. So that's why it is not infectious in that way. We can use in BSL2. Now, as we go to BSL3, more the hazardous potential increases. Okay, so that are indigenous or exotic, meaning that they are new to the environment or can be potentially hazardous in case of respiratory transmission are used in BSL3 environment. For example, like Yersinia pestis or Mycobacterium tuberculosis that are transmitted through respiratory mechanism can be used in BSL3. And the final most is the BSL4 that is really dangerous and infectious as well as communicable. So in that case, they are high risk aerosol transmitted infections that the organisms that cause those infections are uh, included in this particular level, and Ebola virus and smallpox virus are the examples that must be worked in BSL-4. Okay, so any doubts in this case?
సోదిగా
Sorry. If not, then we'll move on to the next question. So the major requirements for a person working in BSL-4 cabinet are as follows. So we have to select what are the requirements in BSL-4 cabinet that a person needs. So is it a PPE kit or the isolated room for cell culture? It's a sterile air shower before entering or leaving the facility, or all of these are needed. Can anybody tell?
Option D.
Okay. Any more
All the
answers? All the above. Yes. So everything is needed in a BSL-4 facility. So starting with the room that we have. So here is a particular picture of the BSL-4 cabinet that the people are working with. So they are wearing a PPE kit, that is personal protective equipment. And as you can see, these are the wires that are connected with this particular instrument, because these PPE kits are really airtight. So we have to maintain a proper flow of air in these cases. So that's why these wires are linked to these particular PPE kits. And as you can see, there is a separate culture room for this particular BSL-4 cabinet, because these organisms that are maintained in BSL-4 are really infectious. So we cannot take any risk for that. And we have to take everything starting from storing to working, as well as the removal of the waste must be maintained in a separate manner as compared to the normal cell culture. So here it will be maintained in a separate room that will have a sterile air shower before entering as well as before leaving the room. Okay. And for that, everything will be different. For example, here this instrument is called as a -80 that we have. So it will be separate for the organisms that we are working in BSL-4 cabinet. And all the incubators as well as the things that we use will be different from the normal cell culture or normal experiments that we do. Okay. So this is the main introduction for the BSL-4 cabinet. Okay. So yes. So what percentage of CO2 is required for growing animal cells? Can anybody answer this?
Option D, 5% CO.
Okay. Any more answers? So yes, the correct answer is option D. And as you know, like there are few reasons that only this much amount of carbon dioxide is maintained in cell culture. Is that first, it like animal cells grow in certain amount of carbon dioxide, because like in the normal body itself, we have certain amount of carbon dioxide. That's why the cells are adapted to that particular condition. And secondly, carbon dioxide is a buffer component that we discussed in the last lecture also. So it has buffering action and it maintains the buffer of it maintains the pH of the particular cell culture environment. That's why only this much amount of carbon dioxide is maintained in the cell culture system. Okay. So anybody has any doubt? not, then we'll move on to the next question. So, what things will be needed to maintain aseptic conditions in cell culture system? So here are the options. Can anybody guess or give an answer?
Option D.
All
Okay.
the above.
Option D.
Yes. So the correct answer is option D. And in order to maintain the aseptic conditions in any culture, being a bacterial, plant, or animal cell culture, so we generally use a sterile work area, the autoclave equipments that we use. So any other thing that we are using, for example, any tips, pipette tips, or the appendix that we use, microfuge centrifuge, or the pipette that we use, we generally autoclave those things and then we'll use for cell culture, as well as the media. Media is generally not autoclaved in case of animal cell culture because certain components, example antibiotics or those things will get denatured by autoclaving. So we generally filter sterilize that media, that particular media for animal cell culture. And before working, as we discussed for the laminar flow hood, that before starting any experiment in the laminar flow hood, we just sterile the area with ethanol and then we switch on UV and then we'll start our experiment after some time. So this is the thing that we use for maintaining the aseptic condition so that any other contamination is not present in cell culture systems. Okay, so let's move on to the next question. So a lady was suffering from breast cancer. The doctors decided to culture the cells and look for any modification in the cells as compared to the normal breast cells. So what kind of culture is this?
Primary cell.
Okay. Any more answers?
Primary cell culture.
Primary cell
Okay.
culture.
Mhm. Fine. So yes, the correct answer is primary cell culture. And this is basically the mechanism that we use for isolating the cells and doing a primary cell culture. Okay, so for example, this is the tissue that we want to culture, the tissue that we want to culture inside the lab. So what we'll do, we'll just chop this tissue and then that is a mechanical step. And after that, we need some cells also out of that. So the tissue comprises of a cell that are maintained with the extracellular matrix. So then we do trypsinization that allows the cells to break off from the extracellular matrix and are separated in the culture. And after that, we culture those cells in a particular media that is different from the normal media that we do during normal cell culturing. Okay, because these cells are directly isolated from tissues, so they require a particular more sensitive environment to grow. So that the composition of the media will be different from those for the normal cell culture that we use. So then we grow these cultures and after some subculturing. Okay, so because these are directly isolated from tissues, they have some characteristics of the cells that are present in the tissue or the normal body. So to adapt them to the normal in vitro conditions, we generally subculture to around 5 times. And then when these cells are like adapted to the cell culture conditions after certain subculturing states, they are transformed into secondary cell lines or the secondary cell culture. Okay. So these are the and after that we when subculture the secondary cell lines that remains as a secondary cell culture because now the cells have adapted to grow in particular in vitro environment rather than being in a tissue or in a live organism. So here the cells are transformed into the secondary cell culture. Okay. So anybody has any doubt in this case?
Sir, if the cells are already differentiated, then how do we make them de-differentiated to grow them in a culture?
Okay, so what happens is we'll require certain growth factors so that they will start dividing into they regain their ability to divide and then we'll start them to so that they can divide and adapt inside the in vitro environment. For example, like there are some factors like growth factors, insulin growth factor or the epidermal growth factor that we give for culturing certain amount of cells. for I think liver cells we give in insulin growth factor to grow in vitro. Okay, so in that case, we are transforming those cells from differentiated to non-differentiated cells. Okay, am I clear?
Yes, sir.
More details will be there when we study more about the different types of cells and how we do culture them. So more details will be there. Okay, so if not, then we'll move on to the next question. So a person seeded HeLa cells for experiment, and the next day he observed that the culture plate had bacterial contamination. So what could be the possible source of contamination? And here are the few options. Can anybody guess?
More than one option is correct.
Okay. Any more answers?
So option D, more than one option.
Yes. So the correct answer is more than one option is correct. So as we know that uh, the hood might not be sterile, as I discussed. Maybe some other person who have worked in the hood before might not have cleaned, or some residual thing is left in that hood. So that can be the case. Or yeah. So option B is another important scenario that happens. So like we generally do in our labs also, what we do is whenever we touch any animals or in any bacteria that we have some experiments with bacteria, we do not handle cells after that, because there is high chance that after handling bacteria or animals, there is some contamination that will be going into the animal cell culture. So after handling anything that is non-cellular or non-animal cellular, that we have to wash everything, or we have to wear new clothes as well as bath sometimes, and then we have to handle animal cell culture. Okay, so this is the common precaution as well as the common way that we use in our labs for handling animal cell culture. And the third option is the media can be contaminated. Yes, this can also be the option, because sometimes the media, the whole bottle of media can be contaminated due to improper handling or improper filter sterilization. So there are cases in that case also. So yes, the correct answer is more than one option is correct. Okay. So yes, care must be taken to handle animal cells, because the the cell culture catches contamination very easily. So we have to be very careful. Even the primary cell culture and secondary cell culture are kept in different hoods, as well as they are cultured in different rooms in some cases also. Okay, so when the primary cell culture shifted to secondary, then we can shift it in a normal hood and we can work in that case. But the primary and secondary cell cultures are separated in that case, because primary cell culture also gives contamination to the secondary cell lines. Okay, so there is utmost care that must be kept while working with the animal cell cultures. So moving on to the next question. So what is the function of the HEPA filter that we use in the laminar flow? So here are the options. Can anybody guess?
Option B is correct, sir.
Option B or D?
Option B, Bombay.
Yeah, okay. Any more answers? So yes, the answer is option B. And here is the basic mechanism of the functioning of HEPA filter. So what happens is it works in a 3-layer format. Okay. So the first layer that we have is the pre-filter. So in this case, all the large dust that we have for the pollen, large dust particles or smoke. So those are mainly stopped by the first layer, that is the pre-filter. And the second one is the filter that have a very small pore size. So that will not allow any of the particular contaminant to pass through it, may it be dust or small size of pollens or any other cells also that are coming from the back air. So the true HEPA filter or the high efficiency particulate air, that is the full form of HEPA. So the second layer depends on the pore size. So it has a very small pore size. And the last one is the activated charcoal layer. So even small bacteria can pass through this, like very small, or this small viruses can pass through this, that are basically captured or absorbed by the charcoal layer. So so after this layer, only fresh air is passed on to the hood. So this is a three-step mechanism that is followed by the HEPA filter. Okay. So are there any doubts in this case? So if not, then this is the last question for today. And if there are any doubts for the lectures that were discussed in week 2, you can discuss that.
Sir, could you please explain once again about the biosafety levels?
Okay. So, so these are the biosafety levels. So we grade it from 1 to 4. Okay. And in the lowest one is the low risk. And as we move to higher biosafety levels, the risk that we are working with the particular microbe increases. Okay. So in case of BSL1, we work with less like the organisms that are found to be non-hazardous to humans or the environment. For example, E. coli or the normal cloning that we do in BSL1 laboratory or BSL1 hood. So the normal microbiology experiments or the normal techniques that we learned in our bachelor's or master's are done in BSL1 hood only. Okay. And all the non-infectious bacteria that we work with come under the BSL1. Now, in case of BSL2, what happens is there is some risk. So they can be potentially hazardous to the environment or the person that is working with it. So we work with some those cases. We uh, like put in BSL2. So, for example, that is Salmonella species because they can be infectious as well as non-infectious. So we don't know what species are we working with if we are isolating some new organism. So in that case, we'll be working with BSL2. Okay. And in case of BSL3, what will happen? Like there are some exotic organisms, or when you know that the organism can cause a particular respiratory transmission or a lethal disease, can belong to the BSL3. For example, tuberculosis, Mycobacterium tuberculosis, because it can transmit from one person to another. That's why we can work with BSL3, but it will not like transmit in a very like what we say, in a very fast mechanism as compared to Ebola or any other viruses. Okay, so that's why it is like put in BSL3. And now when we are talking about the BSL4, that is the highest level, in that case, what happens is there is very high risk of that particular organism to be transmitted through any means. Okay, through aerosol or any other means. In BSL3, we can work with those organisms also that are not transmitted through air transmission. Okay, because then it will be easier for us to carry out the experiments. But in BSL4, as you saw the picture, that how the people work with BSL4. So in that case, we have to take utmost care with working with those infectious organisms. So in that case, like for example, Ebola. So we work with Ebola, or even some labs work here with SARS-CoV-2. They work in BSL-4 cabinets only. Okay, am I clear? Or
Yes, sir. Thank you, sir.
okay. So any more doubts?
Sir, can you explain the 12th question?
Sorry, which question?
12th question.
12th question. Okay. So shall I explain it from the start, or any step you didn't understand?
From the start, sir.
Okay. So, uh, so as you know that the question states that the total number of cells in a culture is counted. When you counted the number of cells in a particular petri dish, they came out to be 2.7 into 10 to the power 6 cells per ml. Okay. So this was done using Tripe and Blue exclusion method, that is used for counting the number of cells that are living or non-living. Okay. So now, as you have to plate these number of cells, so you have to dilute it a particular time so that you can calculate the amount of cells. Okay. And to count the number of cells, you have to dilute it. Okay. So then you diluted the suspension in 27 times. Okay. So now when you diluted it, the number of cells that are present in the particular volume of cell will decrease because the volume has increased of the suspension. Okay. So now when you diluted it, the suspension 27 times, so the number of cells that now the suspension has is the number of cells that were found before into the dilution factor that we had. So now the cells number is 1 into 10 to the power 5 cells per ml after dilution. Okay. Now from this diluted mixture, you have to add particular amount of cells in a 96 well plate. So you added 100 microliter of this suspension to every plate in the 96 well plate. Okay. So now you have to calculate the number of cells that is present in each well of the well plate. Okay. So how will you calculate that? So you have added 100 microliters, and then from there, and you have the suspension volume like this. Okay, so you have to just multiply this number that we had and 100 microliter that you added in the well plate. So after multiplying this, the number of cells that each well had is 1 into 10 to the power 4. Is it okay, or shall I explain again?
No, sir, it's okay. Thank you.
Okay.
So I have a doubt regarding the anoxic cells, because the normal oxygen content of the blood is around 80 to 90. So the 20% oxygen is more of the oxygen in the air. So in vivo, the oxygen content is around 80 to 90%. So how do we correlate that?
So your question is that in anoxic conditions, how is anoxic conditions develop inside our body, right?
Yeah, exactly. I mean, our body oxygen content is around 80 to 90%, whereas the 22% or the 20% is that of the air. So how do we correlate the in vivo 90% with the in vitro 20%?
Okay. So, yes, that's a scenario that we talked about in the last lecture also. So what happens is, like in our body, there are multiple mechanisms and multiple interactions that are going on. Okay. And in case of in vitro conditions that we are working with, we are working, for example, there is HeLa cell culture. So that is a homogeneous HeLa cell culture only. Okay. And like, what we say. So if we are talking about any cell that is present inside our body, there are several mechanisms that are like maintained to to regulate each and every process. Okay. But in case of in vitro cell culture, there are no mechanism to be there for their protection. Am I clear? So what happens is, like in our normal body, the oxygen that we use is diluted out in every step that we see. So for example, there is a cell that is present to the furthermost point, and from the blood, it's taking the like it is utilizing all the oxygen. So from there, the amount of oxygen that is present, or like the amount of oxygen that reaches to the cell is very less. As well as there are all other cells that are also present in that environment. But in case of in vitro cell, there is no other mechanism that will help the cells to protect it from the oxygen. Okay, so they are under direct contact with the oxygen. So that is more harmful for the cells. That is why we are maintaining those particular amount of oxygen inside the in vitro condition as compared to the in vivo condition.
Thank you, sir.
Okay, any more doubts? If not, then we'll close on to the today's lecture and we'll meet you guys in the next week.
Sir, could you share the questions on the portal, please?
Thank you, sir.
I'll upload the presentation into the website, then you can access from there.
Sir, once could you show me the values of the question number 2? I mean, sorry, the question number 3. Question 3.
Which question? Question number 12 or 2?
Question number 3, sir. Question number 3.
3. Okay. Is it visible?
Yes, sir. It is visible, sir.
Mhm.
Yes, sir. It is fine, sir. Thank you.
Okay. So we'll close the session for today. Thank you guys for listening.
Thank you.
Thank you, sir.